Hi Dallas. So AdAlta announced earlier this week that manufacturing's on track. Can you give us any further insight into the current progress?
Yeah, certainly. We're very excited where we're at the moment. We've done our cell line development. We're near the completion of that cell line development, just waiting on the regulatory documentation. So in the next couple of weeks, we're going to be complete and finished with our cell line development. We have other ongoing activities though. We have the purification development, we have formulation development, we have cell culture development. So there's a lot of things still happening and still to happen.
So what do you see as the next major manufacturing hurdle?
I think the next closest hurdle is what we call the confirmation run. So this is the first time we take the whole process, which is the cell line culturing, the purification. We all put this in one run at a 50 litre. It's the first time it's done in its entirety, and we're expecting that to be completed by mid-March.
Okay, so materials available for the tox and phase 1 studies, when are you expecting those?
We're still on track from a timeline perspective and a budgetary perspective. Our tox material, we're expecting that to be finished and ready for shipping end of June. Phase 1 material, December 2019 for a phase 1 trials, early 2020.
And you've got a long and esteemed career in antibody development. Tell us quickly about some of your prior experience before joining AdAlta.
Yeah, look, I've worked with antibody, FC fusion companies, vaccine companies for close to 20 years now. So I've been at AdAlta for 15, 16 months. Previous to that, I was at a small NASDAQ listed company. It was called NextVet. It was solely an antibody FC fusion company. So my duties there were very similar to what I'm overseeing here at AdAlta. So it was all process development, manufacturing, formulation, stability, the whole gamut of product development. So I spent over 3 years at NextVet doing that. Previous to that was CSL. I spent 14 years in a couple of stints at CSL, a lot in the analytic characterization of antibodies and FC fusions, and also some time in just biological purification development at CSL as well. So that just in there is 17 years of working with antibodies and FC fusions.